we've generated this reference for the healthy immune system, and the Allen Institute for Immunology is now digging into several disease states, including rheumatoid arthritis, multiple myeloma, inflammatory bowel disease, COVID-19, and others. It's also important for us just to understand how the immune system works in healthy people in as much detail as possible.
Have you ever heard of the Immune Cell Atlas and wondered what it is and how did scientists create it? Well, listen up. We have an episode of Teach Me in 10 with Dr. Lucas Gray from the Allen Institute. He's going to tell you all about what it is, how researchers can use it, and why it's so important for advancing human healthcare. Over to you, Lucas.
So the Human Immune Health Atlas is a new release from the Allen Institute for Immunology, where we collected samples from 108 healthy pediatric, young adult, and older adult donors that all had no history of any autoimmune disease or any chronic infection, so that we could generate a cross-age atlas of the healthy immune system. So this was work done in collaboration with clinical researchers here in Seattle at the Benaroya Research Institute for all of the healthy adult samples, and with collaborators at the Children's Hospital of Philadelphia and the University of Pennsylvania for the pediatric samples. So this covers 3 age groups: pediatric subjects, younger adults age 25 to 35, and older adults age 55 to 65. And in each case, what we asked our partners to do was collect blood and plasma samples from all of these donors. And we generated peripheral blood mononuclear cells from the blood and profiled the proteins that are present in the plasma from the same visits for each subject. And in addition, we also did a panel of clinical labs, kind of standard laboratory panels for things like blood chemistry, complete blood count, and lipid panels. So we're hoping to bring all of this information together to create a really nice cross-modal reference data set for healthy immune systems. So what we've done here with this Human Immune Health Atlas as a first release is look at single-cell RNA sequencing data collected from all of those samples. So across those 108 subjects in total, we generated a single-cell RNA-seq data set with more than 1.8 million highly curated, really high-quality cells. We then very carefully clustered and labeled all of the cell types in that data set so that we can understand what all the different subsets of cells are across those different axes of age. And built a nice website with a bunch of interactive tools and resources for people to dig into the data. So along with information about the cohorts and characterization of those patients, we have pages on our site dedicated to describing the experimental methods that we performed, the data analysis methods, including the actual code and notebooks for every single step that we took to process that data, and a certificate of reproducibility, which is our framework for tracking steps in our analysis. We also provide all of our cell type definitions, what our expert annotators used to determine what different cell types were and how we describe those. And we also provide some models that other researchers can use to label their own data using those cell type definitions. So if you've got PBMC data from single-cell RNA-seq, and you want to apply the labels that we use, we provide information about how researchers can do that. And finally, we have extensive downloads around this data set. So if you're a researcher who really wants to dig in and use this data set for your own research, we really welcome you to do that. We've provided all of the single cell data. All of those clinical laboratory results can be downloaded now. And soon we'll be adding flow cytometry and O-link proteomics data as well from the same set of samples. So we hope that all of this will be a really great, healthy reference for a lot of different researchers. And that's exactly how we're using it in-house. So we've generated this reference for the healthy immune system, and the Allen Institute for Immunology is now digging into several disease states, including rheumatoid arthritis, multiple myeloma, inflammatory bowel disease, COVID-19, and others. So we're using this as kind of our reference point for what the healthy immune system looks like. And we invite other researchers to use these resources to do the same. So on the website itself, we also have these interactive tools that I mentioned a bit earlier. And we've provided 3 ways so far for people to dig in and start asking some questions of the data. So we have what we call the gene expression reference for the Human Immune Health Atlas, where you can easily just put in a gene that you study and view the expression of that gene across age groups and across all of the cell types in our reference. We also have a UMAP Explorer, so you can look at all 1.8 million cells at the same time, laid out in 2 dimensions using a UMAP projection, color those by all the different features of these subjects, or by gene expression or cell type, to explore the data that way. And finally, since we have all of this great clinical data, we've made a really flexible tool for people to be able to examine and slice that data across any of the metadata that are available for these subjects and samples. So you can really dig in and try to look for things that change clinically over time across many different facets of these subjects. So for anyone wanting to learn more about the data set, in addition to what's present on the Immune Health Atlas website, we have a bioRxiv preprint now under review to describe and go further in depth on what the healthy immune system looks like over time. In this Immune Health Atlas, we're looking at a single time point, but all of these subjects were profiled longitudinally as well. So we looked at 9 or 10 time points over a 2-year period in each of these subjects. And in our preprint and the manuscript, we describe what happens over time, and that allows us to look at other axes of variation and distinguish aging from what happens intermittently over time in the immune system. And with that as well, we also did flu vaccine tracking. So we looked at day 0, day 7, and day 90 for a flu vaccine time course in order to see differences in vaccine response across age and also across things like cytomegalovirus infection status. So CMV is a virus that's found in about half of all adults and usually isn't a problem for us as long as we have a healthy immune system, but it does cause some differences in the composition of our immune system and our immune responses. So we dig in and characterize that in our paper, as well as other changes in things like flu vaccine response. So for us, part of the impetus for creating this resource and creating this data set was to really understand the variation that we see in healthy immunity in order to have that baseline for our studies into disease. So the immune system varies from person to person, and it's important for us to understand what those differences are so that we can really tease apart those interpersonal variations from disease variations. It's also important for us just to understand how the immune system works in healthy people in as much detail as possible, so we can try to just gain more basic understanding of the immune system. And finally, this resource in particular, where we've performed cell labeling, gives us a really great way and a really great tool set to label our disease and other data sets so that we have a consistent way of comparing across all of our data. So when we're talking about a naive CD4 T cell in one data set, we know we're talking about exactly the same cell type or the same population of cells in another. And so we hope that other researchers may want to use and adopt this same nomenclature and same cell type labeling so that we can go across data sets with our labels.
Thank you for joining us over here at Technology Networks, and a big thank you to Dr. Lucas Gray. Subscribe to see the next episode of Teach Me in 10. Bye!